All of the previous studies and their conclusions convinced us that DNA is indeed the genetic material of the cell. But the next question that needs to be answered is, how does DNA pass on the genetic information? One of the keys in understanding how DNA does this is knowing its structure. Now, in 1940s, during the time when Avery, MacLeod, and McCarthy performed their experiment using the R strain and the S strain of the pneumococcus and the proteinase, RNase, and DNase. There was another guy named Erwin Chargaff that performed experiments on DNA samples from several species of living organisms. He hydrolyzed the DNA samples, so he was able to separate the purines, the pyrimidines, and he ran thin layer chromatography on the purines and pyrimidines that he obtained. And these are his findings. Number one, the base composition of DNA generally varies from one species to another, which is somehow aligned with the fact that DNA is the blueprint of life. Because one species will produce the same offspring. It cannot produce an offspring of another species. Chargaff's second conclusion is DNA specimens isolated from different tissues of the same species have the same base composition. So if DNA carries the blueprint of life, then within a living organism's body, the composition of the DNA must not change. Otherwise, there would be some mutations probably or something that would happen in the living organism that, for example, would make it turn into another species. But that doesn't happen. The third, the base composition of DNA in a given species does not change with an organism's age, nutritional state, or changing environment. This again is aligned with the fact that DNA carries genetic information. This observation means that the DNA is preserved in a living organism as time passes by. And the most important of all of Chargaff's observations or conclusions is the final one. In all cellular DNAs, regardless of which species a sample came from, the amount of adenine is equal to the amount of thymine, and the amount of guanine is equal to the amount of cytosine. And it also follows that if you add the amount of adenine and guanine found in a sample of DNA, it will be equal to the amount of thymine and cytosine found in the same sample of DNA. And this is what we call Chargaff's rules. Now, we have already seen that adenine forms 2 hydrogen bonds with thymine, and guanine forms 3 hydrogen bonds with cytosine. Now, these Chargaff's rules is actually very important in the discovery of the double helical structure of DNA, as we will see later on. So as people became curious about DNA being the genetic carrier or the molecule carrying genetic information inside living organisms, two prominent people, Rosalind Franklin and Maurice Wilkins, decided to use a method called X-ray diffraction of DNA fibers. We will not be discussing X-ray diffraction. So we will just discuss the findings of Rosalind Franklin and Maurice Wilkins. So they found two patterns, two repeating patterns in the X-ray diffraction of DNA fibers. The first pattern occurs with a distance of 3.4 angstroms between the spots. And the second pattern occurs with 34 angstroms as the distance between the spots. So these are their major observations. With these, they were able to propose that a DNA molecule assumes a double helical structure. Now, with these structural findings, and together with Chargaff's observations and other studies, two guys, Watson and Crick, were able to come up with a three-dimensional model of the DNA. So based on the structural studies by Franklin and Wilkins, they concluded that there are two polynucleotide chains winding around a common axis to form a double helix. The two strands of DNA are anti-parallel, meaning one strand runs from 5' to 3', and the other strand runs from 3' to 5'. The bases, your adenine, guanine, thymine, cytosine, occupy the core of the helix. So we find them inside the helix, while the sugar and phosphate chains run along what we call the backbone. So we find them on the outside of the helix. The surface of the double helix contains two grooves. We have the major groove and the minor groove. So in this illustration, this is the major groove. This is also another major groove. And this is the minor groove and another minor groove. And finally, incorporating Chargaff's rules, each base is hydrogen bonded to a base in the opposite strand to form a planar base pair. So in line with what Chargaff observed, your adenine will pair or form a base pair with thymine, and your guanine will form a base pair with cytosine. Now, this proposed three-dimensional model of Watson and Crick was able to answer the question of how DNA passes genetic information. So what they proposed is that a parent DNA strand becomes separated. So these two blue strands separate, and a complementary daughter strand is formed. And since we know that adenine will always pair with thymine and guanine will always pair with cytosine, we know that the daughter strands will have the same sequence as that of the parent strand. In addition to these, Watson and Crick also described the sugar moiety of each nucleotide in the nucleic acid as having the 2' carbon in an endo conformation. So again, we have the sugar phosphate backbone, the nitrogenous bases forming hydrogen bonds with their partner in the other strand. The orientation of the planar rings of our nitrogenous bases is perpendicular to the axis of the double helical structure. And we have the minor grooves and the major grooves. With this structure, it was also able to explain the observations of Franklin and Wilkins, the two patterns. The first pattern, which consists of spots with 3.4 angstrom distance, this 3.4 angstroms is the distance between two consecutive nucleotides in our double helical structure. Meanwhile, the second pattern with the 34 angstrom distance refers to 10 base pairs in one complete turn of the DNA. So if you have the first base pair here and go around the DNA molecule and complete one pair, you would have passed 10 base pairs. And the distance of the first base pair to the 10th base pair is 34 angstroms. Now, Watson and Crick also decided that this double helical structure would be a right-handed helix. When we say right-handed helix, the direction of the helix's turn is the same direction of your fingers in your right hand when you curl them. If this is the case, then everything fits the experimental data.